Hello everyone, this is Pankaj Kumar Patel and I am going to present a topic about the basics of enzyme, which is a part of MSc third semester paper biochemistry, and we will discuss about various aspects of enzyme and how basically enzyme work, what is the different mechanism, what is the structure, etc. So let us start the topic.
Now, the learning objective of this tutorial is basically, first thing is we need to understand what is an enzyme or a biocatalyst, an alternative terms used for enzyme. And the second is how actually the enzymes work and what is the underlying mechanism that enzymes use to catalyze any specific reactions. Furthermore, we will discuss few of the enzymes examples in the tutorial. Now, starting with the enzymes, so enzymes are majorly proteins or RNA molecules that are produced by living cell, and they are highly specific to catalyze a specific reaction. That means every reaction that takes place in the body required a separate enzymes. Now, specificity is although concerned with the enzyme, but later on we will discuss about the type of specificities for the enzymes. Now, the enzymes are major macromolecules which basically involved in the biochemical reactions that takes place in the body or in the organ system, whether it is a plant or animal or bacterial or any system. Now, in the most of the cases, the major macromolecule or the enzymes are proteinous in nature. Few of the examples of RNA are as well found which act as enzyme. One of the common example or the well noted example of RNA as enzyme is peptididyl transferase activity. Now, this is the enzymes are the only molecules. Because of that, most of the biochemical reactions could be carried out easily in an organism. Otherwise, the survival of any organisms would be difficult. Basically, what we used is the term biocatalyst we have earlier used. What does it mean? That how it is different from normal catalyst or chemical catalyst. That differ in the catalysis is that the biological product formed or biological substrates are used in case of enzymes. Like chemical catalyst, the biocatalyst as well as could not disturb the equilibrium of any reaction, though they can alter the rate of reaction. Now, catalyst majorly concerned with the increased rate of reaction, but in case of biocatalyst or enzymes, few of the reactions are known in which cases the enzyme or the presence of biocatalyst or presence of enzyme somehow degrade or reduce the rate of reaction. Unlike biocatalysts would not survive in the body for indefinite time. Since these are protein molecule in nature, they get slowly degraded, and because of that, they cannot maintained in the human system for lifetime. So we have to replace, or in the plant system as well, so we have to replace the enzymes time to time. Catalysts normally have a broad specificity, whereas every step of reaction required a different enzyme or a biocatalyst if you consider the biocatalytic system. Now let us understand the structure of an enzyme. So here, as you can see, that I have shown a. So let's see the structure of an enzyme, or the basic structure of enzyme. This is a structure of proteinous enzyme. So as you can see, it's a protein structure that is formed with the amino acid sequence. Now this is a surface model of a protein that if an enzyme exists in the liquid form, it would look like this. Now these enzymes could be formed with a single polypeptide or a multiple polypeptides. Now every enzyme contains various sites. One of the first site or space that contains by the enzyme is known as active site. This is the active site, where the active site is the basically site where the substrate of molecule get attached to be converted into the product, isn't it? So the active site is the primary site of an enzyme which basically involved in the catalytic reaction. Other sites included allosteric regulatory sites. Allosteric regulatory sites or allosteric or regulatory sites are basically sites which do not directly interact with the substrate, but yes, it interacts with the different compounds, and because of their interaction or allosteric site interaction, the activity of enzyme or the catalytic power of enzyme greatly affect. Sometimes binding of a certain specific activator increase the rate of reaction or specificity of the enzymes 1,000 times, whereas sometimes, or the allosteric regulator, if it binds with the allosteric site, it may reduce the enzyme activity. Now in addition to protein part, the enzymes may contain non-proteinous part. So the enzyme, the protein part of an enzyme is known as apoenzyme, and the non-protein part may be a coenzyme, that is an organic part or organic molecule that is required for the catalytic reaction of enzyme, or a cofactor, that is a metal ion. Normally a metal ion is considered as cofactor, whereas an organic molecule is known as coenzymes. So in addition to protein part, there may be a cofactor or coenzyme. We will shortly discuss various coenzymes and cofactors which are associated with the enzymes, and their deficiency may leads to various deficiency diseases. But now considering the point that the protein part of an enzyme is known as apoenzyme. The protein part of any enzyme is known as apoenzyme, whereas the complete protein or the complete enzyme, that is apoenzyme plus coenzyme or cofactor, is known as holoenzyme. And the functional unit of an enzyme or the functional capability contains only by the holoenzyme. The apoenzyme alone could not catalyze any reaction. Now let's consider the properties of a protein. So the protein have a colloidal property or colloidal nature. So enzymes are giant molecules that their molecular weight may ranges from 12,000 to over 1 million kilodalton. So these are colloidal in nature. That means they diffused at very slow rate. And the overall efficiency of an enzyme or the catalytic power of an enzyme also depend on their diffusion efficiency as well. Now the catalytic power, as I told you, is depend on that thing, but the catalytic power of enzyme should be measured, and it is measured in terms of turnover number or molecular activity, that is the activity that is performed by a mole of enzymes. Now continuing to the properties of enzyme, the enzyme have very high specificity towards its substrate. That means every reaction required a separate base or a separate type of enzymes. And the enzymes may have different specificity, that is a substrate molecule, a single substrate may be one specific molecule, that is an enzyme may be act as enzyme for a specific molecule only, or it may have a structural similarity, that is a group similarity. Say for an example, hexokinase is an enzyme which can specifically recognize all the hexokinase or hexose sugars including glucose, galactose, maltose, etc. Whereas some enzymes have a optical isomerism property. That means if a compound possesses optical isomerism, then the enzyme could specifically recognize all its optical isomers and can perform a reaction or can catalyze a reaction. And sometimes what happens that the enzymes is very specific towards a single form of chiral molecule or single chiral form of the molecule, and that is basically a chiral specificity. Now in the active site, the side chains of an amino acids that may include carboxylic group, ammonic NH2 group, CH2OH group, these serve as the catalytic group or active site of the enzymes. So the active site contain this free side chains which basically involved in the catalytic reaction. Now enzymes may require various cofactors. Yeah, that means metal ions like zinc, magnesium, iron. Like catalase is an enzyme which required iron, whereas a few of the photosynthetic enzymes required magnesium. Now enzymes of DNA polymerization required zinc as well as copper in some cases. Likewise, even the magnesium. So likewise, specific enzymes required a specific ions for their proper functioning or for their catalytic power, and that ions are known as cofactors. Now few of the enzymes required complex organic molecules like biotin. Biotin is necessary for carboxylation reaction. Now additionally, like NAD or NADPH or FAD, these are also complex organic molecules which required for dehydrogenation or dehydrogenase activity. So few of the cofactors I have listed for the lecture, like copper is required by cytochrome oxidase assembly, whereas iron also required by catalase or cytochrome oxidase assembly. Potassium ions required by pyruvate kinases enzymes. Likewise, magnesium, molybdenum. Molybdenum is required by dinitrogenase. And dinitrogenase is the oxygen sensitive enzymes which is used for nitrogen fixation. Likewise, zinc, nickel and different ions including calcium, these ions are required for the catalytic power of the enzymes. Considering the coenzymes as well, these are complex protein molecules, complex organic molecules like coenzyme A, which is made up of pantothenic acid, and its deficiency may lead to dermatitis. Likewise, various organic molecules including FAD, FMN, NAD, thiamine phosphatase, cyanocobalamin, pyridoxal phosphates, which are produced by vitamins. And that's why the deficiency of vitamin leads to deficiency diseases. So what is the vital role of vitamin? Is they are the part of enzymatic machinery, and these enzymes are quite necessary for the proper functioning of the system, organism's system. Now these are the few examples of coenzymes as well. And these coenzymes and cofactors get binds to the apoenzyme and form a complex holoenzyme, and holoenzyme is the functional enzyme. Now how this holoenzyme or enzyme work, or how it basically enhance the rate of reaction. Now consider a hypothetical equation or a hypothetical reaction in which a stick has to be bent and broken. Now, a stick may be bent and broken into two pieces without any enzymes or in absence of enzyme. Like if you someone eat food, or like the every organism required energy. In that case, for the digestion purpose, enzymes are required. So enzymes basically break the long chain carbohydrates or proteins or fatty material. Same as a stick. A stick may be broken into two parts. Likewise, a complex molecule may be broken into various parts in presence and absence of enzymes. But enzymes, why do we need enzymes? Because in absence of enzyme, the digestion of normal humans' food required 3000 years approximately, and that takes place in few hours, like 48 hours or 24 hours in the presence of enzymes. So here a stick has to be bent and broken into two parts, and that required, this is the initial energy of stick, say for an example. So the energy level should be increased, or energy should be given to stick to bend it and break it. Now after breaking, the energy would be reduced. Why? After breakdown, del G, normally what a reaction takes place where del G or change in the free energy is always negative. So Gibbs free energy will reduce after breakage of this stick. But see, for the breaking as well, even though the del G is negative, for del we need to apply certain amount of energy. Now if enzymes bind with the stick and changes its conformation, it form a product, then what happens? Basically enzyme substrate complex formation takes place, and that gives energy to the stick. The exact way of enzyme working is, another thing that happens is a transition state. See, we have three stages involved here. A stick, it gets bent and broken. So if you use an enzyme which has a conformation of transition state, like it, that is bad stick. So when it binds, the enzyme bind with the stick, it automatically make the stick to bend, or make stick to bend, and that making a stick to bend increase the energy of a system. That is, ES complex has some level of energy, and this energy may be used to break down the stick. So by providing energy to the substrate, on binding with the substrate, enzyme and substrate binding release certain amount of energy, and that energy would be utilized to break down the substrate into product. Or if substrates binds with the enzyme that leads to liberation of some amount of energy, and that energy may be used to join the two substrate and form a product. So it may happens in two ways. That is, a substrate may be broken into product, or two substrate may be joined into a product. But in both the cases, enzyme has to be bind with the substrate, and the enzymes normally possess a conformation of transition state that leads to accelerate the rate of reaction. Now we have discussed a term, that is enzyme efficiency, or enzyme activity may be measured in the K catalysis. The unit that is used to assess the efficiency of an enzyme is known as K catalysis. We will discuss about K catalysis, about enzyme inhibition, about the internal mechanism of enzyme efficiencies in the next tutorial. But for the tutorial, you may understand the thing little bit like this, that the enzyme efficiency or the catalytic power may be defined in terms of K catalysis. That is, K catalysis is always per second, and K catalysis may be roughly known as turnover number. That is, number of product that is formed by from the substrate by the enzyme in a given unit of time. So here you can see catalase may catalyze like 40 crores, I think. Yes, 4 crore of molecules per second, whereas Rec-A protein and ATPase may catalyze 0.5 molecules per second. So every enzyme has its own efficiency to perform a work, and normally the anabolic enzyme have lesser efficiency. Anabolic enzymes, that means where an assimilation of energy into chemical energy takes place, is basically they have a lower efficiency, whereas the catabolic enzymes normally have a higher turnover number. So this was about the enzymes and basic introduction of enzymes, and we have discussed, let's have a quick recap, that we have discussed about what is enzyme and what are the structure of enzymes, what is cofactor, what is coenzymes, and how it basically works. We have discussed about theoretical enzyme stickage, and then we have discussed enzyme efficiency as well. Now, to assess, let's have a quick assessment that chemically enzymes are what? RNA or protein, or both are protein and RNA, or carbohydrates. The second question is whether an enzyme always possesses an active site, a allosteric site, a regulatory site, or all of the above. Always possess. And the answers are, see, chemically enzymes are both protein and RNA. I have told you in the beginning of the lecture itself that most of the enzymes are protein, but few of the enzymes are RNA in nature. And for the second question, it is the active site that is always possessed by the enzyme, or an enzyme always possesses a site is as active site, whereas enzyme may possess allosteric regulatory sites. So the correct answer is active site. And these are the further readings. If you want to elaborate your knowledge, you may please refer the instant notes of biochemistry or Lehninger principle of biochemistry. These are the also references for my lecture. Thank you, and